Greetings, everybody. The COVID-19 comorbid disease cancer is one of the most complex and fatal diseases in the world due to possible metastasis and mutations. In 2018, WHO reported that cancer is one of the leading causes of mortality and diseases in the Philippines. In addition, Alzheimer's, a neurodegenerative disease, is considered to be a global epidemic affecting more than 47 million people. Through faulty genetics and senescence, the risk of obtaining cancer in Alzheimer's skyrockets. Thus, cancer and AD are one of the major challenges of the 21st century that have to be addressed. Multiple reports show that the use of natural products is an attractive strategy towards this. Which is why I, Delphine Inigo Pilapil IV, your speaker, and my co-authors are proud to present our study of the discovery of not a single, but a dual disease-targeting phosphodiesterase inhibitors from Ovaria alba DCM sub-extract with anti-cancer and anti-neurodegenerative therapeutic properties. The cell cycle is tightly regulated by different signaling pathways. One key regulator is phosphodiesterase 4, which is responsible for the hydrolysis of CAMP. However, the downregulation of CAMP has been implicated with different types of malignancies, including colon cancer, leukemia, and lymphoma. In addition, the enzymes USP14 and KEAP1, which play an important role in inflammatory response, are also interesting drug targets for the treatment of cancer. Aside from its role in cancer, phosphodiesterase 4 have been linked to the amelioration of the neurodegenerative process caused by the downregulation of CAMP that has been associated with Alzheimer's. On top of that, acetylcholinesterase that hydrolyzes acetylcholine also plays a role in inducing Alzheimer's. Thus, phosphodiesterase 4 and acetylcholinesterase inhibition hold promise in the treatment of neurodegenerative disease and inflammatory diseases. The family Annonaceae has been found to possess secondary metabolites that have therapeutic properties. Given the pharmacologic profile of the genus Ovaria, we are particularly interested with an endemic Philippine medicinal plant, Ovaria alba. Thus, the main goal of the study is to extract, fractionate, and chemically profile these secondary metabolites of DCM sub-extract from Ovaria alba and evaluate their biological activities through in vitro and computational simulations. My discussion will now be divided into 7 phases. We start from the extraction and fractionation of Ovaria alba, followed by inhibitory assay versus the phosphodiesterase 4, preceded by chemical profiling of the bioactive sub-extract. We then move towards virtuous computational process with their key findings. Anti-cancer and anti-neurodegenerative assay, along with their virtual computations and analysis, will then follow. Finally, I'll be discussing the pharmacokinetic analysis of the active compounds. The leaves of Ovaria alba were collected, ground, extracted with DCM methanol, and concentrated to yield the crude extract, which underwent fractionation in increasing polarity to afford sub-extracts, namely petroleum ether, UAP, DCM, UAD, and N-butanol, UAB. These sub-extracts were then subjected to anti-phosphodiesterase assay. AMP concentration was quantified using a phosphodiesterase kit, and luminescence was measured and results were reported as IC50 values in microgram per milliliter. The DCM sub-extract was the most biologically active and consequently became the area of focus. LCMS analysis was using an impact-2 Bruker HR LCMS QTOF identified 18 secondary metabolites typical of the Annonaceae. To further solidify our findings in our in vitro assay, we use in silico procedures. In our virtual computations for all ligand-receptor interactions, we use Chimera along with Autodoc Vino softwares for molecular docking. Molecular interactions were provided by the BioVia software, and top ligands per target enzyme were subjected to molecular dynamics using AmberMD. The 18 compounds were subjected to in silico simulations, first against PDE4B2. And interestingly, grandifluorescein and cyatostamine had the best binding energy score of -10.2. Having a better pharmacokinetic profile, cyatostamine was subjected to further in silico analysis. The complex was stabilized by pi-to-pi interplay occurring in 4 regions of the molecule and hydrogen bonding between AsP392 and chromanon core. Furthermore, RMSD and RMSF analysis showed dynamic stability for the complex over time. Next, Anti-proliferative and cytotoxic properties of the plant samples were assessed using a cell titer blue assay. Assay was carried out using healthy HUVEC cells, K562, and HeLa cancer cell lines. GI50 and CC50 scores indicate that the UAD had the best anti-proliferative and cytotoxicity properties. The 18 compounds were now docked against USP14, a deubiquitinating enzyme. Dicamenitin exhibited favorable binding towards USP14 with binding energies of -9.8. Post-doc analysis revealed that there are two prominent stacked pi-to-pi interactions responsible for dicamenitin's attachment towards the binding site. It is further stabilized by pi-to-pi and pi-to-alkyl interactions. MD computations show its stability through time. Molecular docking was also performed for KEAP1, and its structures is divided into 4 discrete binding domains. In this study, we are particularly interested with the C-terminal Kelch domain brick-a-brick BTB domain. The Kelch domain serves as a binding site for NRF2, affording cytoprotective mechanisms. Dicamenitin exhibited the best binding energy of -11.1 against the KEAP1 Kelch domain. Its attachment to the domain's active site is primarily through H bonding and pi to sigma interactions against the chromanone core. Similarly, MD computation showed the complex stability within the given time frame of 50 nanoseconds. The central BTB domain is another important region of KEAP1, particularly for its role in providing sensing mechanisms to oxidative stress. This domain houses the nucleophilic residue cis-151, which was found to be one of the most reactive residues in the regulation of cellular stress. Molecular docking results revealed that grandifloracin had the best binding energy of -7.3 toward the KEAP1 BTB domain. Grandifloracin interacted with cis-151 through its tricyclododecanone core and one of the benzoyl substituents via the pi-to-alkyl interplay. The resulting complex is stable as per molecular dynamics simulations. Interestingly, the top ligand for this enzyme possesses alpha-beta unsaturated carbonyl group. These compounds fastening such electrophilic moiety have grown interest as drug candidates because of their ability to interact with nucleophiles of enzymes such as cysteine residues, potentially through Michael addition. We then calculated the frontier electrophilicity potential of the metabolites through HOMO-LUMO energy calculations. Grandifloracin showed the highest electrophilicity index and kinetic stability. As such, grandifloracin was used as a Michael acceptor to model the Michael addition of cis-151 of KEAP1. DFT calculations revealed that the mechanism involves the formation of an enol intermediate through concerted addition followed by keto-enol tautomerization to yield the final cis-151 grandifloracin keto adduct. Lastly, to further solidify the application of secondary metabolites against neurodegenerative disease, acetylcholinesterase inhibition was evaluated using a slightly modified LMADS assay. UAD was the most biologically active, and thus its compounds were further subjected to molecular docking. Docking results show that dimethoxychalcone as well as practilactone had the highest binding energy of -9.8 against ACE. Having a more favorable pharmacokinetic properties, dimethoxychalcone was subjected to further post-doc analysis and molecular dynamic simulations, where it was revealed that the ligand is nestled to the enzyme's binding site in a stable conformation. The top-ranked ligands of bractilactone, grandifloracin, cyatostamine, and dicamenitin was predicted to have good gastrointestinal absorption and poor blood-brain barrier crossing capacities. In addition, they have druggable properties and are non-toxic. To conclude, Uvara alba DCM sub-extract exhibited potential anti-cancer and anti-neurodegenerative properties. Via LCMS, we identified 18 putative secondary metabolites from this bioactive sub-extract. To further explore their biological activities, in silico simulation against several drug targets was conducted. Lastly, this is the first study to highlight the Michael addition reaction of grandifloracin with cis-151. For future prospects, we recommend the isolation of the aforementioned compounds for confirmatory in vitro and in vivo testing. I would sincerely like to thank the IFS for funding our study and my co-authors for giving me this opportunity. Thank you once again and have a blessed day.